Hello, dear colleagues, we are starting the publication of our new online intensive Maturation Management. This program will be more relevant for those who keep caviar broodstock in permanent fattening temperatures, i.e. in the RAS or on warm water farms. At the same time, some aspects, such as optimization of feeding parameters at different stages of maturity, may also be of interest for sturgeon farming under natural temperature regime. Growing sturgeons at permanent fattening temperatures has proven to be an excellent method of producing meat sturgeon, but when transitioning to caviar production, a diametrically opposite situation is often observed. Instead of accelerated maturation, as was the case with linear growth, it is often noted that the maturity stages, especially the end of the second stage, are accelerated and, consequently, the overall duration of maturation is increased. Often, females of the same age group mature in small batches over a period of 4 years, and the total maturation time sometimes exceeds this figure when grown under natural temperature regime, and the result of the maturation is not always pleasing. Whether these processes can be controlled and what methods of impact on the reproductive cycle can be used, all this is discussed in the series Maturation Management. First, as usual, the theory, and this time it will be a bit more than usual. Traditionally, we try to avoid detail on anatomy and histology, but not this time. Basic information about the processes occurring in the gonads, in our opinion, is essential and gives a key to understanding the sources of possible problems. Therefore, we will start by discussing what happens to the gonads during the maturation process. Indifferent or juvenile period of gonad development. At this stage, the gonads are laid and formed, and associated changes in the structure of the germ cells occur. As a result of mitotic divisions of primary germ cells, smaller cells defined by the term gonads appear. After a series of mitotic divisions, gonads of various orders appear. The gonads of the last orders are either oogonia or spermatogonia. Signs of anatomical differentiation of the gonads toward the ovary or testis are not detected during this period. Visually, the gonads at this stage are thin white-pink or white-yellow stretches, a few millimeters wide, which, due to their small size, are not visualized by ultrasound diagnostics. After the appearance of adipose tissue in the gonads, depending on the species, approximately in the 5th to 8th month of life, the stage of gonad differentiation begins. According to available data, the differentiation process can begin at different amounts of adipose tissue in the gonads, which varies significantly depending on age, species, and environmental conditions, and can range from 40 to 90% of the gonad volume. The first stage is characterized by an increase in the number of sex cells, which occurs due to mitotic division. There is no increase in the size of the germ cells at this stage. Anatomically, this state of gonads in sturgeon occurs only once in a lifetime during the first cycle of maturation. All subsequent reproductive cycles begin with the second stage, but the processes of mitotic division of germ cells inherent in the first stage continue throughout life. This is not a separate state of juvenile age. At this stage, changes in the anatomy of the gonads that appear to be typical of one of the sexes become discernible. In females, such a visual indicator is the fissure groove, which runs along the entire gonad on its lateral side. The development of the gonads in the female direction is most pronounced at first in the caudal part of the gonad, i.e. just where the amount of adipose tissue is the least. By the end of the stage, the formation of ovipositor plates on which the oogonia are located begins throughout the furrow, in a perpendicular direction to it. Future eggs are transparent, irregularly angular in shape, tightly adhering to each other at the edge of the ovipositor plates. Male gonads at this stage are distinguished by the localization of the generative tissue, which is also located on the lateral side of the gonad, but on the outer part of the fatty tissue base. From this stage, gonad development can be monitored by ultrasound screening, since both the gonad as a whole and the generative tissue are clearly visualized on sonograms. As shown in the slide, dark anachogenic fat occupies most of the visualized area of the gonad. The ovarian generative tissue is visualized as a gray hypochogenic stripe with jagged edges and is surrounded by fat both above and below. The generative tissue of the testis is tightly adherent to the abdominal wall and is visualized as a smooth stripe that is darker than fat, with a well-defined smooth lower edge with a bright hyperechogenic band formed by the testis sheath. It should be noted that differences in the dynamics of gonadogenesis in sturgeon species and hybrids with different maturation rates are well manifested already at this stage of maturity. The slide shows sonograms of gonads of 1H sterlet, left, and Russian sturgeon, right. It is easy to see that sterlet yearlings, despite their smaller mass, have better developed gonads characterized by more intensive development of generative tissue. Maturity Stage 2. This is one of the longest stages of gonad development, which can last up to 3 years. In first-time maturing fish, depending on the species, Stage 2 begins in the second or third year of life. In rematuring fish, the second stage begins after spawning, with the beginning of the fattening season. At this stage, ovaries acquire yellow coloration, testes, milky white or with a slight pink tint. Growth of eggs throughout the second stage occurs not due to the accumulation of nutrients in the oocyte, but due to an increase in the mass of cytoplasm, cellular structures, and venom of the oocyte. Therefore, this period is called the protoplasmic growth period. Due to the intensive growth of eggs, the size of which increases several times, there is a significant increase in the volume of generative tissue of the gonad, and to ensure this process consumes a significant amount of protein entering the fish organism with food. As we have already said, at the second stage, the anatomical structure of the ovary begins to form. The stroma of the gonad becomes multilayered, egg-bearing plates are formed, which are flat outgrowths hanging down into the body cavity on which oocytes surrounded by follicular cells are located. Oocytes or eggs become clearly recognizable to the naked eye, acquire an oval shape and are located tightly to each other in the middle of the egg-bearing plates. Oocyte development is uneven, and even by the end of the stage, several generations of eggs of different sizes and phases are distinguishable from those at the very beginning of the protoplasmic growth period to those already completing it. At the cellular level, the protoplasmic growth stage includes 3 phases: early, middle, and late, corresponding to the beginning of the middle and end of the second stage of gonad maturity. On the slide, we see a schematic representation of the changes occurring in the oocyte as it passes through the second stage. As the oocyte grows at the very beginning of the protoplasmic growth period, cellular elements appear in close proximity to the nucleus, then move into the cytoplasmic thickness. This is the end of the trial fragment. The full version of the intensive can be accessed on our channel on the Boosty platform. You can see the address of the internet channel of the Sturgeon School and our email now on the screen, and they are also given in the description to this video. Thank you for your attention. We thank everyone for their participation and invite you to our other online intensives and workshops.